Recording in progress.
Okay, so I'm Jean Grassman, and along with Susan Fountain, we're the Watchdog Health and Safety Coordinators. So we'd like, first off, just thank you for your interest. Your interest means that you care about your workplace, you care about the conditions you're working in, and by extension, you know, that of your coworkers and your students. And so it's really good to see you all here with this interest. Although, you know, it's a little perverse because we really wish we didn't have to meet and talk about mold, but we do, and that's the topic for tonight. So this is also a part of a series that the Watchdogs are doing this fall. As you know, this is being recorded and it will be available on the PSC website. We also have done sessions on workers' comp and long COVID, and advocacy by using PESH. So we're trying to develop a library of topics that, you know, we all run into all the time. So with that in mind, I'm very pleased to have Ed Olmsted here. He is a friend of the PSC. He is a long-standing industrial hygienist, and he has his own consulting company where he is very busy and very in demand. So having him for a couple hours is a special treat. I also can say that Ed used to work at NICOSH, and we know about NICOSH from our Thursday Watchdog meetings, which are from 3:30 to 4:30. You can see that on the PSC calendar. And he worked at NICOSH years ago, and I have to say, I went to one of his presentations given by NICOSH probably when NICOSH was in Midtown. And so I'm guessing it's like 20 years ago.
A long time ago. That was when they were on
time ago.
7th Avenue and 26th Street over
Yeah.
there. Yeah.
And really, I came away from that session, and I'm an industrial hygienist, but I got my orientation in you know, and what it, you know, basically how mold occurs and what you need to do to mitigate it that evening from Ed. You know, so my schooling didn't really do what Ed was able to do. So with that in mind, I'm going to turn it over to Ed, and he's going to tell us a little bit about what we can do with what we face as the mold problem in our workplace.
All right. Well, let's see. I'm going to bring up share this screen. You should be able to, let's see. All right, so I think everybody should see now my slides coming up, hopefully. If not, let me know, but this is the I started out just to show you just to give everybody some samples of reference material. That's an AIHA journal. That's the second edition of their journal, their manual that Dr. Ling Hung put this together, or she was the editor. I've worked with her on a bunch of projects. I just finished a project with her down in North Carolina a few weeks ago. So she and I work together from time to time. And this is a good reference manual. It really goes, it gives you everything you would want to know about how to do assessments, how to inspect buildings, you know, how to deal with hidden mold, how to deal with what kinds of samples you take, how to evaluate, and ultimately it goes through how to mitigate and remove and abate and, you know, bring the building back to something close to what it was. It's never possible to fully clean up a building once you get it really water damaged, but you do the best you can. So that's a good reference manual. This is the second edition. It was published, I want to say in 2018, maybe somewhere around there. And there are others. This probably, you know, obviously you would have to go to the AIHA website and you can order it through them. but I would say that's the best reference manual, but you have to pay for that one. There are ones that are reference materials that you can get that are you don't have to pay for, that are free, and that's some of the stuff put together by EPA, right? And so they have published some material. It's not going to be nearly the kind of depth of information that you would get from the AIHA stuff. It's really for mostly the public, where it gives you some sense of what the hazards are and how to deal with it and how to clean it up. But it really doesn't really get you much on give you much on the industrial hygiene aspects. When I say industrial hygiene, I mean how do you collect samples, how do you take tests, and how do you figure out whether you have a problem. Generally, in most cases, EPA and New York City Health Department and New York State, they usually advise people that for a moldy building, you don't really have to come in and take lots of samples. And that's true, but it's not always true, in that sometimes you have situations where it takes a little finding. The mold isn't necessarily going to grow where you can see it, or it doesn't necessarily grow to a degree that you can see, but there's still a problem. So, New York City and their regulatory agency, the HPD, which is the Housing Preservation, when they come in and do an assessment of somebody's apartment, if you call in and complain through 311, their criteria is entirely visual. That's all they do. They'll look for visible water damage or visible mold, but they don't go any further than that. So I'd say that they catch maybe 70% of the problems. Of many buildings, the problems with water damage and mold is hidden, and so you won't see any of it, and that does let a lot of things fall through the cracks. And unfortunately, landlords are kind of aware of how HPD operates, and so to fix a problem, sometimes you'll find a landlord will just paint it over, right? So if you have a wet, water damaged wall in your living room and you call 311 and HPD comes in and issues a citation to the landlord, it's not unusual for the landlord to go and paint over that damage and then invite HPD back. And since they can't see any mold or water staining, they remove the violation and they consider it cleared. And many times I come in and have to argue with a judge in housing court that all they did was paint over it, and if we cut a small hole in the wall, we can find the mold is all growing inside the walls, in the wall cavities, and that can still present a problem for people's health. So it is possible, and it is useful to base your surveys on visual, and you can root out at least a good fraction of the problem sites, but it's just relying on visual evidence you're not going to catch everything. So that's why with the industrial hygiene manual, we have all of these different kinds of sampling methods to try to get information on the things that you can't see. So again, molds are microscopic, right? They're microorganisms. So by definition, you can't see them all the time. And so sometimes the growth is not sufficient for you to see it. And most times it's hidden. Another bit of reference material, this is more on the health aspects. So this one I think came out somewhere in I want to say around 2010 or 2012. I'm not certain, but it looks more into the health effects of damp and moldy buildings. And there are other guidelines. This is free also. It's a CDC NIOSH document. And the World Health Organization has quite a few things, reference papers, and you can get those from their website. They have some things on mold. So there's a lot of free info out there. So getting to what the issues are, right? And the problem when we have damp, and I should take a step back and say that when looking at the health effects, both the World Health Organization and the National Academy of Sciences, they're the Institute of Medicine. They looked at all of the reports and data out there and tried to come up with an analysis of the state of the art of reference materials as it applies to moldy, damp buildings. And so if those documents are also available free, and essentially what they're saying, what they've concluded is moldy, damp buildings can cause upper respiratory symptoms, allergy symptoms, sinusitis. They can cause an increased rate of sinus infections in people who are living in those conditions. And those where there's a causal relationship. They then go on to say that there's an association between being in a damp, moldy building with onset of asthma and more problematic respiratory symptoms. But that's an association. So there is, at this point in time, there is not a direct link between people developing asthma and being in damp, moldy buildings. It hasn't been proven. So the health effects that are widely accepted, and where there is a strong indication that moldy buildings, damp buildings can cause symptoms, and those are what I mentioned. It's the sinus problems and the upper respiratory problems, more allergy-like symptoms, but it doesn't have to be allergy. We'll go into that a little bit. So generally, what happens in damp buildings is people are going to start inhaling mostly particulate matter. Some of it might be organic compounds that the fungi and that the bacteria produce, but to a large degree, it's this dust or particulate matter that gets generated because there's so much biomass developing, right? As these organisms grow in a wet area, they're reproducing and you're generating a lot of junk, mass, material. The molds are particularly a problem because their life cycle is to produce spores. So when molds produce spores for the purpose of reproducing, right? So when a mold spore, which they always bring this up, and lawyers always bring this up in court in their defense, mold is everywhere, and to some degree that's true. Molds and fungi make up a fairly large fraction of the biomass on the planet, right? So they are everywhere, growing mostly outside. And spores are everywhere. Spores settle out outside, they settle out inside. There isn't a surface that you could test in most buildings where you won't find some spores, right? The more dust you have on a surface, when you look at a surface of a shelf or a cabinet or the top of your monitor on your computer, and you see that dust that's settling on there, that's what we would call house dust. Probably some fraction of that is going to be fungal spores. Well, fungal spores would include different kinds of spores. Molds are considered fungi. Fungi include more than just molds, right? Fungi include mushrooms and puffballs and yeasts, and molds are just one part of what we consider the kingdom of fungi. So they are eukaryotes, so their biology is closer to ours than it is to bacteria. So they're more like us, and which does make it hard to deal with when you have an infection from a fungi, because their biology is not so different from ours. So to kill them, to find a drug that kills them would be harmful usually to us. It's not like bacteria, which have very different biology, and you can find ways to thwart them that doesn't bother us. Anyway, so what happens with molds in particular, they're sort of a unique aspect of fungi, is they produce spores like most fungi do. those spores are their reproductive propagules. That's how they reproduce. So those spores are designed to move with the air. They're aerodynamic. They're usually, depending on the species, mold spores can be anywhere from 3 to 5 microns in diameter, like aspergillus spores or penicillium spores, or they can be much larger. So there's a pretty wide range, but all of them are, I would say you could probably put them all together somewhere between 3 microns and 20 microns is what might be typical. 20 would be a pretty big spore. It's starting to getting into the realm of pollen. But so spores come in various different species, produce different size. They're all microscopic, so you're not going to see them. So and the purpose of those spores is to get released into the air, and they travel with the air currents. So I always say, when I'm testifying in these cases, is mold spores go where the air goes. So wherever those spores are released that get into the air, they'll travel with the air. So if you have a lot of mold in your basement and it's producing a lot of spores, mold's growing in the basement, producing lots of airborne spores, that air is going to travel up into the building, and the mold spores will travel with it. So that's, kind of keep that in your mind. Mold spores go where the air goes, and it can travel from inside wall cavities into buildings, right, into the living areas. So it's not as easy because air doesn't easily go from a wall cavity into your house, but it goes to some degree, right? Air does travel from outside, even when you have all your windows closed and your doors closed. The air is going to find its way in, right? And the spores, or some spores, are going to travel with it. They can travel through electrical outlets. They can travel through wall switches, light fixtures. So there are pathways. It's possible to wall them in, but you have to make it a very good air barrier. But so that's an important thing. The second thing that's important with molds is that, as I said, they produce these spores for purposes of reproducing. So if you look at, and I think I have some pictures, if you look at growth of molds, you'll see that what the main thing they're doing once they've reached a certain phase is just to make spores. They become a spore factory, and they're just putting those spores up into the air. And so when you have an apartment that's water damaged and there's mold growing on the walls, that mold growth is going to keep putting spores out into the air, and the concentration in air is going to go up 10-fold or 100-fold or more. Suddenly you've gone from 100 spores per cubic meter in a normal apartment, and you're up in the 10,000 spores per cubic meter. So the dose goes up. People tolerate mold spores pretty well. We breathe them outside all the time. But the problem is when you have these very high concentrations, it starts to induce some inflammation in the respiratory system, which is where we're mostly impacted. We mostly are inhaling these spores, and I've put in there dust, mold related dust, and hyphae are the fragment, are the hyphae and mycelium in mold growth is the vegetative growth. It's not reproductive. It's not spores, but I'll show you some pictures of that in a few minutes, and you'll get a better sense of it. So just to kind of give you an overview, the molds produce spores. Those spores go wherever the air goes. They settle out of the air at some point. Takes a long time, hours. They settle onto a surface, and the spores are very hardy. They can live for 5 or 10 years, just sitting on a dry surface. They're waiting for water, and when they finally have some water that presents itself, it takes about 24 to 48 hours for that spore to germinate. So a lot of times you'll read on the EPA website or somewhere else that mold growth will start in 24 to 48 hours. That's true. What they really mean by that is the germination process starts in that amount of time. just having that spore turn into a growing mold is not necessarily in and of itself very problematic. So we can have growing mold, vegetative growth on our wall that's wet, and not really have it present much of a risk to us because it hasn't reached a point in its growth cycle where it's actually producing spores. The point when it becomes a health risk, or when our dose or our exposures start to go up, is when this growth reaches a phase where it's producing spores or it's reproductive growth structures. And so I'll give you an example. If you have a wall that's got green mold growing on it, penicillium, let's say it's penicillium, it looks kind of blue-green in a lot of cases. You may have seen that in your refrigerator for those who have left an orange in the bottom of the refrigerator and forgotten about it. And suddenly you see it back there and it's covered with a green powdery moldy cover. That's penicillium. Usually that's Penicillium chrysogenum. It grows very nicely at cold temperatures. It likes it actually prefers that cold temperature in the refrigerator, which is kind of nice because if you accidentally ate that orange, it probably wouldn't survive very well in your stomach, and it certainly does not. A mold that likes that cold temperature doesn't like our body temperature. So we'll get more into infection and things like that in a little bit. So anyway, if you were to take that green mold that's growing on that orange and cut about a square inch of it off the orange, so now you had a square inch piece of visible mold. In that square inch, you probably would have in the order of 1 to 10 million spores. So that's kind of, even though you can't see them, that's a heck of a lot of spores in a small area. And so you can imagine what happens when you have an entire wall with visible mold growing on it, how many trillions of spores you have being released from that wall. And if you're standing in that room, you're dosed to these mold spores that you're inhaling now, because again, they float in the air and they move with the air, including into your lungs. So you're really upping your dose. So that's our main route of exposure. And I put there not just spores, but also if you're disturbing this stuff, if you're cutting the walls, if you're demolishing this moldy wall, you're generating fragments of this biomass. And the biomass is not just going to be spores, it's going to be all of these vegetative hyphal structures, hyphae and mycelium. They're filaments. So the mold spore germinates and starts shooting filaments into that material that it's wet. And those filaments are living, I guess, roots, you could almost think of them as. They're vegetative structures, and will grow wherever the water is. So it will grow in that material, and it uses the water hydraulically to actually push these filaments through this wet material. And it will exude enzymes into whatever it's growing in, and those enzymes will break down to a large degree. Let's say it's a paper sheetrock. It will break down that cellulose into sugars, and then it will reabsorb those sugars, and that filament or hyphae, it then will move that water mixed with sugar throughout the colony. So it can it can move these nutrients all over this growing colony, and it's making all that sugar into energy, right? Just like we eat food to make energy, they're going to absorb those digested sheetrock paper to produce energy for themselves. And the more energy they can produce, the more filaments they will grow, and life is good as long as it stays wet. They'll reach a phase, and I find most of the time, depending on what material they're growing in, if it stays wet for 1 or 2 weeks, in that range, you reach reproductive structures start getting made. Right? So just like all other living things, when life is good and you're getting lots of food and you're getting water and everything's going fine, you start thinking about reproducing, because that's the end game for all biological organisms. So it's going to get there in 1 or 2 weeks. Some materials it will take longer, but in a piece of sheetrock, it will certainly be after the first week of being wet, there's a pretty good likelihood that you've reached sporulating mold growth. It's now has these mold spore producing structures, fruiting bodies. As a mushroom is a fungi, a little different, not a mold. It doesn't reproduce quite the same way, but when you actually see a mushroom, the bottom, the gills of that mushroom, the bottom of the cap is where all those spores are, and it's producing those spores also to germinate and grow somewhere in soil. It's possible to be exposed by ingestion, so you don't want to eat moldy foods. There have been cases of farm animals that have gotten sick from eating moldy hay or moldy corn and things like that. And some of these molds will produce certain toxins, right? And so there have been cases of farm animals being dying and getting ill from eating moldy corn and ingesting aflatoxin, which is what the Aspergillus flavus produces. We'll get a little bit more into toxins. I don't want to dwell too much on those. But that is the third bullet there. Sometimes people can react and have skin reactions. Sometimes that's just an allergy. It's possible if you're handling without gloves, if you're handling very moldy materials, some molds do produce toxins, which we call mycotoxins, which might be absorbed in through the skin. These are not very, in my experience, the mycotoxins, it's hard to know how much of an effect they have on people's health in moldy buildings. My own sense is, to a large degree, it's just inhaling all these dust and spores, and that's leads to inflammation of the mucous membranes in the airways, and that chronic inflammation, because you're living there for weeks and months, that chronic inflammation can lead to bronchitis and sinusitis and these kinds of things. This is what I've been talking about. This is what some sample from a wall that has some visible mold on it. When we look at it under the microscope, this is the kind of thing you'll see, lots of spores. Here we're only seeing spores, and this is a genus called Eulocladium. So you can have a shot at identifying what mold you have by the shape of the spore, but not terribly, only the genus. Here's a little better shot. There's a different kind, that's Chetomium. And there you see the little straight filaments there, those are the mycelium or the hyphae. And they're kind of broken up. This sample just got this particular genus is a type that produces spores in what are called an ascus, which is a big sac. So it fills this sac up with spores, and then if the sac ruptures, it will release all those spores into the air. But it's the same kinds of function and properties. It's going to grow these vegetative structures first, this mycelium. It's going to break down what it's growing in and start absorbing the sugars. That it uses to make energy, and if life keeps being good, it will then make these spore producing structures, and that's Chetomium growth under the microscope, and that's at a magnification of 1,000 times, so they're tiny. Most of the time when we worry, as I said, we worry about molds, is we worry about the inflammation effects. The breathing these large numbers of spores in all day for weeks and weeks will lead to your immune system being more and more sensitized and triggered by this, right? So our immune systems, the more you expose it to a certain protein, we've just gone through all this with COVID, right? You put mRNA of the virus spike into a person's blood, right? And the cells will produce the spike protein, and then that will trigger an immune response. And if you do it again, you get that sensitization effect happening, that the immune system has a memory, and it keeps getting better memory, and it keeps building up a better response. So the immune system will get more learn as it goes. So it will get more specific, and it will respond more and more the more you're exposed. That's the adaptive immune system. That's the part of your immune system that produces antibodies. And the molds will induce an antibody production, but mostly what we call IgE, and it's a allergy type of antibody. But it can induce an IgG, which is a serum type of antibody. So molds, if you're exposed long enough, can induce an antibody response through your immune system. But the primary effects, and I think it's not entirely all been sorted out, but the primary effects of breathing in a lot of mold spores and a lot of mold junk is your immune system also has a innate branch, right? So our immune systems have an adaptive part and an innate part. The innate part starts working from the day you're born. It's you breathe in bacteria or some mold spores, and they have various kinds of molecules on their cell walls or on their cell membranes. Molds have beta glucans and some other kinds of molecules that we don't have in us. So our immune systems have developed and learned how to respond to those. We have what, I don't want to get into the weeds with immunology, but there's this whole area of what's called toll-like receptors. One of those receptors will respond to fungal pathogen-associated molecular patterns, is what they would call a PAM. So our cells in our body can detect broadly the presence of these foreign mold-associated molecules, and that triggers a cytokine response, right? You remember hearing about the cytokine storm with COVID, but it's a cytokine response where we start releasing all these immune system signaling molecules, and that's what starts to trigger the inflammation. So most of what happens with people living in damp, moldy buildings is they have this innate response, and they develop an inflammation of that area that keeps getting hit by all the stuff they're breathing. And so there is a threshold where what's in the air doesn't seem to trigger all this. It's sort of below the radar. But when you have really high doses, it starts to then trigger an immune response, an innate immune response, which leads to inflammation. And that inflammation would typically happen in the respiratory system, because that's where the spores are hitting. And so you get this inflamed tissues in your sinuses, and you get inflamed tissues in your airways and lungs. And you get the person out of that environment and eliminate that level of exposure, and the inflammation will go down. A typical pattern, though, in a person who's in a moldy, damp building is the inflammation. They keep going there every day, right? They either live there or they work there. And so this inflammation just keeps happening day in and day out and day in and day out. So they're constantly having these symptoms. Oftentimes they'll go to a doctor. The doctor will look in their sinuses or do some other tests and see that there's a lot of inflammation. Sometimes they'll give the person at some point, because if the person's having trouble with chest tightness and cough and really suffering a lot from this, a doctor will prescribe them, if it really looks bad, if their airways really start looking inflamed, they'll prescribe them prednisone or some other steroid, or nasal type of steroid, to help to break, these steroids, these corticosteroids, are these off switch for inflammation. So if you have inflamed tissue in your sinuses and you pump in some Flonase, this corticosteroid, into your sinuses, that will turn that that will turn that inflammation off. And so it's a very effective medicine to help have people feel better. But unfortunately, it masks the problem because they feel better, but they're still breathing all this stuff in. And so it's a temporary fix, and ultimately you have to get the person out of that environment if you're ever going to have a chance of them getting better.
Ed,
yeah.
I wonder if I could ask a question that came up in the chat, since you're
Sure.
talking about this exposure through breathing. The question is, would the blue mask, I assume the surgical type mask that a lot of
Yeah.
people have been wearing since COVID, would that help prevent mold-related breathing problems?
I think it might help a little bit, but so when we do mold cleanups, when we have a worker that goes in and starts cutting out sheetrock, we usually require them to wear a half-face respirator with a HEPA or N100 type of filter on it. It's almost the same kind of respirator you'd wear for asbestos or lead or silica dust or things like that. So I think the surgical mask would reduce your level that you're breathing, but the problem is that it's a very much a sliding scale, and so some people it might work for, but some people who are very sensitized. And what I mean by that is sensitizers are what that indicates is if you develop an immune response to something where your body recognizes whatever this coming into your lungs is not you, it's made up of proteins that's not self, right? That's how our innate immune system is really looking at, is this a molecular pattern from some other organism, like your immune system can respond to what are called endotoxin, which are in bacteria cell walls, gram-negative bacteria, and all it has to do is see some endotoxin and it triggers this inflammatory response, and these chemokines and chemotrans signaling compounds, and that leads to inflammation. In gram-positive bacteria, it will see these peptidoglycans, which are what make up the cell wall in gram-positive. And for fungi, the patterns are of beta-glucans. So if you keep breathing those kinds of things in, you're going to keep triggering this your immune system to induce an inflammation. But what also starts to happen is your immune system will become more sensitized to those proteins. It will react quicker when it sees them, and it will react at lower and lower concentrations. In other words, it might take you breathing in a high dose of, let's say, 100,000 spores per cubic meter for you to have an inflammatory response. But as you keep being exposed over and over, you might start to lower that threshold. So with sensitizers, we can't establish very good threshold limits that will protect people like we can with chemicals, right? So if you look at most chemicals, there are published threshold limits that basically say if you can keep your exposure to that chemical below this limit, you're not likely to get sick. Immune system type of illnesses, molds, and other types of even chemicals and other things that really trigger our immune response, it's a sliding scale. So a person becomes more and more and more sensitive to it, and that means their symptoms can become more severe over time, and their sensitivity, their trigger, their dose, their permissible limit, their threshold gets lower and lower and lower. So you might find a person who at first could tolerate fairly high doses, but as they're exposed, their tolerance gets lower and lower. And then some people reach a point where they're so sensitized that they can't even walk into a place that has even small amounts of mold growing somewhere in it. They cannot tolerate. It's like they get more sensitive over time. It's sensitizing. So a surgical mask might work for a lot of people, but it's not necessarily going to work because it is only lowering the dose, and it might lower the dose enough for most people, but it might not lower the dose enough for somebody who's been sensitized. In which case, if you're very sensitive to mold, you would probably need an N95 kind of mask where you've been fitted for it, and you know that it does a better job at filtering out, right? So an N95 would reduce your dose by at least a factor of 10, whereas a surgical mask maybe not so well. But not a bad thing to wear a surgical mask if it works for you. But if it's not working, then don't do that. I just listed these molds up here because some molds do cause infections, most do not, right? And I think the main reason that most do not is almost most of these molds are soil dwelling. They live outside. They've evolved outdoors, and their job in the nature is to break down dead stuff, right? They're saprophytic. They like to start growing on dead leaves or dead vegetation or anything that's died out there, a tree, and they break it down into they digest it. So they've adapted to an environment that's outside, which is to say they mostly are adapted to rather cold temperatures, right? So most of the species of Penicillium, Cladosporium, a lot of those types of molds are what we call psychrophilic. They like cold temperatures. Penicillium prefers it to be somewhere between 60 maybe and 75 degrees Fahrenheit. And so they don't really, they're not as likely to cause infections in us because they don't do well at 98 degrees. So that isn't, I don't want to suggest that that's always the case, because some molds are weird little critters. They can go, some species are what we call dimorphic. They can go from being a mold, a filamentous type of mold, and under certain conditions, they can become a yeast, right? And I'm trying to remember which species specifically, but so you can have a species that's Penicillium, which is a filamentous type of mold that doesn't like it lives outside. It likes cold temperatures generally. It doesn't really cause its infection. But if you breathe the spores in, it can actually switch to becoming its anamorph, its other form, which is a yeast, and that yeast can actually then turn out to like hot temperatures. It's thermophilic. So some molds, very rare, but some are able to switch. Generally when that happens, it's usually when it's in the form of its yeast counterpart that it can cause us infection. So, there are various yeast-like fungi that are opportunistic, Rhodotorula and histoplasmosis when it infects a person, it's in the form of a yeast when it's doing that. It's not the case with Aspergillus. You see that second bullet, Aspergillosis. That is a filamentous fungi mold, and it actually is thermophilic. It likes hot temperatures, so it will grow and compost, and it certainly grows well at 98 degrees Fahrenheit. So most times when you're in a hospital setting and a patient that has an immune system that's not very functional, maybe they're an organ transplant patient that's taking various medications to lower their immune response so that they won't reject this new organ. So they're giving all of these drugs to lower their immune system. When you're doing that, if they're exposed to too much Aspergillus spores, they could actually develop the spore could start growing in their lungs, in their respiratory system, and it'll grow into a ball of, just like you would see mold growing in some kind of food in your refrigerator. You see a fuzzy black ball. It will grow like that inside the person's respiratory system. So aspergillosis is one of the things that is a concern in hospitals. So that's why when you have a chemotherapy patient or a transplant patient, you put them in rooms to recover. They're in a room that's maintained at a fairly sterile condition. It's not totally sterile. Their patients' room though would have HEPA filters for ventilation and generally limited visitors, and they do things to really keep their exposure to any mold spores to a minimum. And mainly, if they do get an infection, it's going to be aspergillosis. And aspergillus is really the primary one. The histoplasmosis is more of a infection from molds that grow in bird droppings. So those are those situations where you have a construction crew doing demolition and there's a lot of bird droppings in the building or bat droppings. They can stir up enough dust. If you get a high enough dose and the person has maybe something that might reduce their immune system, right? There's a lot of immune compromised people out there in the world, right? If you have type 2 diabetes, your immune system is somewhat reduced. If you're taking various kinds of medications, if you have an autoimmune illness and you're taking TNF inhibitors or things to reduce your immune response, you're going to be more prone to these opportunistic. We consider aspergillus an opportunistic species. In other words, it doesn't infect most of us. It won't infect it. You can breathe Aspergillus flavus and fumigatus or Aspergillus niger all day long. And for most people, they're not going to develop an infection. It's the folks that have something wrong with their immune system. And then Cryptococcus is one associated with birds. And another building-related infection would be Legionella, Legionnaires. That's not a mold. I just throw that in there as another kind of infection. Just that's a bacterial. Generally, the health effects are sensitization, hypersensitivity. What that term means is some, usually in workplace exposures, but it can happen sometimes to people in their own residence. I very rarely, once in a while, I'll get asked to go look at a place that somebody's developed hypersensitivity pneumonitis or allergic alveolitis. What happens there is, and it's not exactly clear why, but the person develops a level of sensitization that their lungs start to develop scar tissue. So with hypersensitivity pneumonitis, the really important characteristic is the person, as the disease continues and as they continue to be exposed, they develop scar tissue in their lungs. Right? It's called farmer's lung because it happens mostly in agricultural settings, where the doses to these kinds of fungal spores is really, really, really much higher than anywhere else you'll get. I've done sampling at for fungi in chicken production facility, egg production facilities, in pig farms and compost facilities, various different kinds of settings where there's really remarkably high levels of spores in the air. Occasionally, though, I do see people's residences. Sometimes I actually see where a place that a person's living that's so moldy that the levels that they're inhaling start to become comparable to what you would see in a pig farm, which, I don't tell them that, but it can happen. Sometimes in residential areas, there's so much mold growing, there's so much water damage that the person's exposure is really quite remarkable. And of course, they get sick often from that. Not everyone does, by the way. It's hard to predict who will get sick, but if you expose a large group of people, you'll probably find a dose that will get almost everyone sick. So it's really hard to predict what level to use as a threshold. Everybody has it's a sliding scale, but if you put everybody into a damp basement that's got mold everywhere and you let them live there for long enough, pretty much everyone's going to find their level that makes them start having symptoms. As I said, it's an innate immune response. There's an inflammatory response. There's these PAMs or PAMPs, pathogen-associated molecular patterns. These are things that trigger the inflammation, like beta-glucans in molds, endotoxin in bacteria, peptidoglycans in bacteria, and they trigger these immune response. It's not a IgE response, and it's not an IgG response. It generally doesn't have to include antibodies. So it's hard to measure clinically. And then there is allergy. That's the most common thing. IgG, when their immunoglobulin, the E, IgE goes up, that's an allergy response. So some people develop allergies, not everyone does. And the allergies they become sensitized. In other words, the more you're exposed, the more sensitive you get. Oh, let me just move on to the next one here, because so again, I think we'll move on from the health effects, right? That's just a little bit on asthma. Again, as I said, there's an association between people working in damp buildings and moldy buildings and the onset of asthma, but it hasn't been fully proven that it causes asthma. There's an association. And asthma, of course, is a reversible airway constriction, right? As the person is exposed to these triggers, it can induce their airways to constrict, and it makes it very hard to breathe, right? There are cases of people dying from an asthma attack. Generally, most people who have asthma would have a rescue inhaler where they will inhale a medication that opens up the airways, and they'll also often take a medication that reduces the inflammation. So they take usually two kinds of inhalers: a rescue inhaler when they have an episode, and an anti-inflammatory inhaler like a steroidal type of inhaler that cuts down the inflammatory response. And they keep taking that until they get their asthma under control, and hopefully they get it to a point where they're not having asthma attacks. The thing about a person having frequent asthma attacks is it can remodel their lungs and sort of start causing some scar tissue and damage. So if you have asthma, you don't want to be in a moldy building where you're having frequent asthma attacks, because each asthma attack leads to a little more damage in your lungs. You want to avoid asthma attacks, aside from the fact that they're really unpleasant. All right, so I think we'll move along from most of these health effects issues. I mentioned the mycotoxins just very quickly. Molds will produce various metabolites, and some of them are toxic. The more infamous that you read about is Stachybotrys chattarum, and that produces a toxin called satratoxin. Their toxins are called trichothecene groups of toxins. There are other molds that produce those kinds of toxins. The likelihood is that these species produce these toxins in order to give them a competitive advantage, right? So if I'm Penicillium chrysogenum and I'm starting to grow and metabolize and breaking down and digesting an orange in the refrigerator, life is good. I'm growing all over this orange, but just like everything else, when something's good, there are other competitors, right? So there's lots of bacteria that would also like to eat this orange, and probably other things. So other microorganisms. And so it's a contest. And the Stachybotrys, or the Penicillium chrysogenum, we all know, in order to give itself a competitive advantage against bacteria, which is its biggest competitor, it produces penicillin, and that will kill the bacteria. So these organisms are just producing, probably, my sense is they're producing most of these mycotoxins because they help them compete against other species. So many species of Penicillium produce mycotoxins, many species of Aspergillus, right? Aspergillus flavus produces the toxin aflatoxin, which, Aspergillus flavus is a soil-dwelling species, and it becomes a problem in the growing of peanuts, because peanuts grow underground, and if it's a drought year and the peanuts are not as resistant, they're not so hardy, they can become colonized by Aspergillus flavus, and so then you can find that the peanuts will have a higher quantities of aflatoxin. So when you go to a baseball game and you buy a bag of peanuts and you're eating those roasted peanuts, most of the time you're fine, but when you get that rotten one, spit it out. Don't swallow the rotten one. That's probably been already colonized by Aspergillus flavus. The good news is that most of the companies that make peanut butter and peanut oil go through a lot of effort to test their product to make sure there's not too much aflatoxin. They do allow some, and aflatoxin is a carcinogen, right? It's so people who work, last I read, people who work in the peanut industry and peanut factories have a a higher rate of liver cancer compared to what you would expect, and it's probably to some degree due to the aflatoxin that they breathe. So I don't know what purpose the aflatoxin is serving for Aspergillus flavus, but I'm sure it's the same thing. It's just competing. So there are many other, Aspergillus niger produces some toxins, and a lot of them do. It's not clear how dangerous that is for those of us breathing them. It's probably, you would have to have a fairly high dose. And Chetomium globosum, that bottom one, also produces toxins. I don't know, I don't tend to get into testing for mycotoxins. Some people go out there and do it. I think it's a kind of a waste of time, and so does the AIHA. So testing for mycotoxins is probably not a very useful endeavor. We'll skip that one. And so in bacteria, right, you have, instead of aflatoxin, we have endotoxin, which is really just a molecule in the gram-negative state of a cell wall. And it can, as I said before, it can induce an inflammatory response. It's one of these innate triggers, innate immune system trigger. All right, so in a building, how do we know when we have a mold issue? There's lots of things we do. So obviously the easiest is to it's water damage, right? Molds, as I said before, these spores need to be present, which they generally are. And they get everywhere by just through the air currents, but they could be tracked in. So if you're, generally, if I vacuum a carpet in a clean house, in a place that is well maintained and has no water damage, I will usually find somewhere between 200 and 500 spores of different kinds per square foot in that carpet. So that's a pretty good amount of spores. And they're probably getting there mostly by settling from the air, but they'll also get there by tracking in, because when you're walking around outside, the soil, there's a lot of soil dwelling molds, and those spores will get, just like the soil and dirt gets on your shoes, some of that is going to be spores. When I take dust samples from high dust, oftentimes we sample high dust what I'm meaning by that is I sample dust up on the tops of door frames, or up on the tops of framed pictures on the wall, or tops of the shelves, or from upholstered furniture. We vacuum that dust. That dust is going to represent more of what's settling out of the air and not what's getting tracked. So I know some industrial hygienists like to vacuum carpets and culture that out and see what is there. I prefer sampling high dust, so I'd rather not have to account for what people are walking and tracking in. So generally, when we vacuum up this high dust, we culture it out and we see what kind of species they are. And we know in most house dust that I sample, I find it's going to be in the amount of spores will be in the trace amounts, right? For typical house dust, just to give you a sense of it, when we vacuum dust off of a high shelf, it will be dominated by cellulose. Is one of the more and that's our paper pushing world. Plant matter, right? Trichomes and other plant matter, pollen. Skin cells is a really big fraction. So skin flakes from the people living there. And synthetic fibers. Those are kind of like the four. When I look at dust in somebody's house, that's what I want to see, the most common prevalent, those four things. When the dust isn't, when their dust is dominated by other things, then I know there's something going on. That's why is it settling out of the air? Usually we find fungal matter, spores, and other fungal structures in the less than 1%, but it's there, and so it's always there. I don't think I've ever seen a dust sample from somebody's house that didn't have some fungal material in it, fungal matter. So we look at how many spores in a certain area, and then also, more importantly, we look at what species are present there. And so certain species of mold are the ones that colonize wet buildings. In a dry building that's never had a mold problem, the types of species we see in the dust are almost always what drifts in from outside. And those are the dominant ones outside are the phyloplane molds, the ones that grow out on the trees, and that's and the mushrooms. So when we culture dust, we often find cladosporium in a healthy building would be the dominant spore that we find in the settled dust. If on the other hand, I go and I measure somebody's carpet in a damp and water damaged house, I might find that I have 2,000 spores per square foot. And I might find that it's all dominated by Aspergillus niger. And that would tell me something's not normal here. So what I want to see is something like 200 to 500 spores per square foot dominated by cladosporium. That would be right in the normal range of what we would see in dust. So you can evaluate what's going on in somebody's house or building by looking at the dust. And I think you can do a better job with that. You can get better information with that than taking an air sample, right? Because air samples, as probably some of you might know, certainly the industrial hygiene folks would know that the air samples have a lot of possibility for error, right? So the air samples are a useful tool. I don't think you want to throw out using them, but you have to be careful about how you interpret the results, and you can often get false negative results with an air sample. And dust samples also have some error built in. What you find when you do mold surveys is sampling dust and sampling the air is helpful, useful tool, but can get it wrong. So you never try to become too confident about those kinds of samples. But those are the things we look for. Is there damage from water? And the thing that you can do is use a moisture meter, which is a very useful tool. And essentially a moisture meter, you can probe areas that you think water is coming into a building, and you can compare that to the same materials that you are pretty confident is not getting water in, and then have a sense of do I have moisture? And if the moisture meter is saying you have some measurable moisture in the wall, and then you have you can conclude you have an active water intrusion of some kind. You have water getting in, and the water could be coming from a roof leak. It could be coming from masonry, exterior masonry leaks. It could be leaking around flashing on a window. It could be leaking from a vent pipe leak, a chimney, or pipes above the ceiling, inside the wall might have a leak. Air conditioners can have leaks. There's we all know there's many places that water can leak, and evidence of visible water damage and measurements with a moisture meter are the two ways to see if you're getting water in. And I said before that water is the key ingredient for mold spores to germinate and start growing into vegetative growth and then sporulating growth. How long that takes depends really, as I said before, it would take a week of something being very wet for that entire week. But it could be a different process. It could be there's a roof leak and the material stays wet for 3 days and then dries out. So the spores would have germinated and maybe started producing vegetative growth, but then as it dried out, they go dormant and they just sit there. And so they'll sit there and not do anything. Once it's dry, it becomes dormant. But then the rains again a week later and the roof leak kicks up again, and that material gets wet again, and it stays wet for another 3 days. And so wherever that growth left off, it will renew. It will start where it left off. The stuff that was there and dormant kicks back on. And so now it starts to grow more hyphae, more vegetative growth, and it keeps growing until that dries out. And then you get the idea in an iterative process. It wet, dry, it goes from active growth to dormant, active to dormant. And it will reach a spore producing point in its growth cycle at some point. It might take longer. So when we flood a building and it's, there's wet walls, we can get mold growing on those walls if it stays wet for a week to 2 weeks. But if we have intermittent water damage and it gets wet for 3 days and then dry for a while, then wet for 3 days, it might take 3 months or 4 months to start getting mold to reach a point where it's producing spores. And once we have that growth, it's very hard to just clean it off. A lot of the custodians in our schools and a lot of the supers in our buildings and maintenance workers in various other kinds of buildings will often be under the impression that you just need to go and throw a lot of bleach on there and that will take care of it. And that just, it doesn't work very well, because when you clean, all you're really cleaning is the outer surface. But so for example, if I have mold growing on a wall, it will grow through the wall, and more of the growth will be on the interior of the wall cavity than what's on the outside. So as I said before, it's going to be, the hidden mold is going to make up much greater amount than what you can see, which is one of the shortcomings of a lot of the standards out there. So we look for water damage, moisture meter readings. We obviously, if you have visible mold growth, then EPA and New York City Health are absolutely correct. You don't really need to test it. If you can see it, it's a problem. Sometimes we test it to make sure that that's actually mold growth. That happens a lot when you have a supply air ventilation diffuser, right? The air conditioning system up there on the ceiling, you can see that diffuser. They often get a lot of dirt around them. And people think, is that mold or is that dirt? And it's hard to tell, and so we test it. And you look at it under the microscope and you can tell it's not hard. It's really an easy analysis. You can tell whether those are spores and hyphal fragments and conidiophores and various other fruiting bodies. You can tell really easy. It's not rocket science. I get into court a lot and everybody gets appalled by the fact that I look at my own samples. Oh my God, you can't imagine what they all react like I'm some kind of, the next step for me is murder. But it really isn't hard to do that kind of a microscopic analysis. It just confirms what your eyes are already seeing. If you're seeing mold growth, that's a pretty good indication there's a problem. I just happen to like to look at whatever that mold growth is under the microscope to be certain, and because I actually like looking at mold samples under the microscope, but they're pretty interesting looking things.
Ed, I wonder if this would be a moment to ask, there are a couple questions in the chat about testing and cleaning.
Yeah.
Okay, so let me just go back to there were actually a couple more, but I'll get back to those hopefully before the end of the session. So let me just start with the testing and cleaning ones. One question was, how do you send mold for testing and where do you send it to?
Good question. Well, there are labs, the American the Industrial Hygiene Association, the AIHA, accredits laboratories to do mold sample analysis. So if you want to try to find a lab that can look at mold samples, you would the easiest thing is to go to the AIHA.org, to their website, and they'll list out labs that are accredited to analyze samples for mold. So, and there's many out there, just like, so there's a bunch right here in New York City that are accredited for analyzing samples for mold. New York State Department of Health will accredit laboratories for doing environmental sampling also, but they haven't got a program for mold. So if you go to the ELAP folks in Health Department, New York State Health, they haven't started accrediting mold labs, but AIHA does. So you would go to AIHA to get a list of the ones in your state or in your city that are accredited. You really do want to use an accredited lab, just so that you know that when you have AIHA accreditation, that means they're going to send you what they call a PAT program. It's a proficiency in analytical testing. They send the lab blind samples, samples 4 times a year, every 3 months, and they have to analyze those and get them right. So and they inspect the lab. So using an AIHA accredited lab gives you some level of assurance that there's somebody's checking and making sure they know what they're doing. When I take, yep, next question.
I wanted to just add on to that. Could you say just a little bit about, you're talking about getting the samples tested. I think the other piece of it is acquiring good,
Yeah.
how to acquire samples in a way that's valid too.
Sure. So, well, in New York, if you're going to take samples and do a mold assessment, New York State is now got a program of licensing assessors. They call them assessors. So if you want to just take a sample for your own interest, the easiest kind of sample, if you think it's mold and you can see it, there's probably the very easiest ways to test. I would take a tape lift, which is clear Scotch tape, put it against that black stuff that's on the wall, and then I put that on a microscope slide. But of course, not everybody has microscope slides. So the other easy way is to either take a piece of that. You can cut a little sliver of that wall. You don't need more than a half a square inch, and just put it in a Ziploc bag and send it out. That's a bulk sample, and the lab will look at it under the microscope and tell you if that's mold growth. Sometimes you can have them culture it and they'll tell you what species are there, but it's easier to just have them look at it under the microscope. Or you can take a Q-tip, and just rub that Q-tip on that black stuff till you get it all on the Q-tip at the end, and just put that in a Ziploc bag, and you can send that to the lab and they will look at the Q-tip. But what you want to have the lab check, what you want the lab to do is what we call a direct exam, a microscopic direct exam, where they are able to identify the presence of spores, hyphae, and conidiaphores, or fruiting bodies. Too many labs out there are just going to give you a spore count. And there's a lot of even some of the AIHA accredited labs, I find, are just doing a really weak job of the microscopy. You want the lab to be able to identify the presence of spores, vegetative growth structures like hyphae, and also conidiaphores, the fruiting bodies or the reproductive structures. You want to know the presence of all three, because if it's the reproductive growth that's important, that's what's putting the spores into the air. So that's the easiest way. And we went around in one of the schools last week, I guess it was, and took about a good 12 or 15 tape lifts from different places where you could see the mold growing, and just sent out to the lab. The tape lifts are and if you are this is something you really want to do, then you can order specialized tape lift type samplers that make it easier to collect a sample, and you can get those from the various catalogs, SKC. But if you're just a homeowner and you want to see if you actually have mold growing in your basement, I think the easiest thing is to use a Q-tip, rub it on that stuff, put it in a Ziploc, and ask the lab to do a direct exam for the presence of fungal structures, and you want them to list out the kinds that they're seeing. So there are many other kinds of samples. Any other questions?
There is a question about, is there a simple, quick test to check for mold that is not high enough? I think not high enough concentration is what is meant there, not high enough to be visible.
Yes, the tape lift can, the lab will give you a report back. Usually all the labs will have a scale. So they'll tell you usually they rate it from 1 to 4. The lab I use goes from 1 to 4. 1 being very, they'll say we have Aspergillus spores, Conidiaphores, and hyphae at a rating of 1, which is to say very light. And if it's a rating of 2, it's a little more dense. I find that usually you'll get a rating of 1 when you can't see it. So, but it's there. So that happens. It's not unusual to have where you take a sample off of a water-stained, water-damaged wall material or something else, and the lab comes back and says you have the presence of Aspergillus growth and or penicillium growth, and it's rated at a level of 1. You wouldn't see that. Now it's a good question, because sometimes the direct exam can be really hard in certain materials. For example, if your carpet gets wet, all of us have had a soaking wet carpet, maybe not all of us, but you've probably come across a wet carpet that smells musty. You can take tape lifts off that carpet. It's very hard to get to pick up the growth structures. So sometimes I will, for a carpet, I actually will vacuum that. Remember we talked about this before. I know what my experience with vacuuming a carpet, that if I have a square foot, I should have somewhere around 200 spores in that square foot to maybe 500. If there's mold growing in the carpet and you vacuum that square foot, you'll actually find anywhere from 1 million to 10 million spores in that square foot. So it's really dramatic. Once you've got stuff growing, the spore concentration just amplifies really, really rapidly. So we have these different methodologies. We can take a bulk sample is to actually take a piece of the material. You could take the backing of the carpet, cut a little piece of it, send it in, and they'll culture it, and they'll send you back results in colony forming units per gram of carpet. I find with bulk samples, if you're getting over 10,000 colony forming units per gram, it's growing in it. So that you can take a sample of a carpet, a wall, ceiling tiles, any other kind of porous material that's gotten wet and you think has mold growth, you can send a piece of it in and culture it. You just have to have a lab that's able to do culturing. I have to say, most of the reports that I read from people doing this kind of surveys, most of the assessors are not doing this, and so they're basically doing mostly just air samples. And I would say that air samples are useful, but it's not it's in and of itself, it's not a very conclusive methodology. It's helpful and it's useful, especially if the mold is so hidden inside walls or under floors that you've just got no chance of getting an actual bulk sample to send in. So the types of samples I have listed here as bulk. I think I have another slide that where we'll go into more different kinds of samples. Well, I'll list them out right now. It's like you can take air samples to see if mold is growing inside the walls, under the floors, or growing in your bedroom or living room. You can test the level of spores in the air in that room, and you take a sample outside, right, as a comparison. And when you compare indoors to outdoors, what you want to see is the indoors typically will have a lower level of spores, and the kinds of spores will reflect outside. That, which is to say, probably mostly cladosporium and maybe some mushroom spores, some basidia spores. Those are going to be the predominant ones. And if you have mold growing in that room, your air sample will become more dominated by Aspergillus penicillium, Chetomium, Eulocladium, these kinds of species that don't show up outside much. So you can sample the air. That's one way to get some data, some information. It's not conclusive. The only real conclusive evidence of an actual mold growth is to take a sample, send it to the lab, and have them tell you that's mold growth. When a lab says it's mold growth, which is to say they would look at it under the microscope. So moisture, these are just other examples. I think we'll just go through some slides. But before we do, in terms of regulations that are out there in New York City, right, we have Local Law 55, which was enacted in 2018. And it's pretty good. I think it mostly covers residential. In fact, it does cover residential, but it's basically enforced by HPD. And these are some of the important requirements. You can download it. It's not that long to read, but these are the, I think, helpful pieces of it. So any HPD will cite a landlord if they find a condition of mold growth, right? If they see mold growth, they look for visual evidence. So they don't sample. So they have to see mold growth, and they'll cite the landlord. But they are cognizant of the fact that you can have mold growth in the ventilation system and inside wall cavities. So that's a little harder for them to pick up. But then lately, now they've added this second piece, the underlying defect. So what they're saying is that we don't have to just find visible mold. If we find evidence that you've got water coming in, right? So if we see water staining and water infiltration and leaks and warping and all that, and efflorescing of that in and of itself will get you a citation as a landlord. So HPD now is basically, since they don't really sample, they either will see mold, which I always find interesting because I don't know how they get away with this in court when nobody's ever asked them, well, how do you know that's mold? Well, how do you know it's not dirt? But apparently it's never come up. Just to show you why lawyers need to get more science and biology training, because it seems like an obvious question to me. But they don't sample it. They just say, we saw it. It looked like mold, so it is mold. But they'll also see water damage, and if they now the law allows them to just cite straightforward water damage. So that's that standard covers preventing asthma. And they also require landlords to inspect their own buildings once a year, all apartments, for evidence of water damage and insect infestation or mice or mold. And they have to inspect upon a complaint. So it's pretty good. I think there's some pretty good, it's harder to enforce because, as I said, HPD does not take samples. So if the mold is not visible and there's no visible stains, it's going to be hard, right? I've seen apartments that got flooded where all you see is a slight warping of the floor, but there's no visible mold and it doesn't look bad. But the mold is all growing on the bottom of the wood, on the underside of the wood floor, but you can't see it, and so you would not get a citation there. You can only tell if that's going on by taking some kind of sample. Either an air sample or vacuum the floor, or lift up a part of the floor and sample the underside. But it is helpful. It is helpful because if you have a lot of water leaking into your building and you call HPD, they will cite the landlord just for the presence of water leaks under this statute. And they will have to fix it. And if you can get the landlord to fix the water leak, you're 90% of the way to getting this fixed. Because you take away the water and then you just have to get rid of that mold growth. Then there's a oh, did I not put the label, the title? So there's a local law 61. Yeah, I guess it's hidden up here. Right, local law 61 is also a New York City law, and that's enforced by New York City DEP. And basically what it is that if in fact you do have a mold problem and you have to abate it, either because HPD ordered you to as a landlord, or you went and had it looked at and the assessor said, yep, you got 100 square feet of mold here, you have to file that job. The contractor has to be a New York State licensed contractor to do mold abatement. The assessor has to be a New York State licensed assessor. They have to file the job with DEP, right? So if as long as there's more than 10 square feet of an abatement, and if the building has more than 10 units, so they won't enforce this against homeowners. But the contractor, the licensed contractor, will file the job with DEP that they're doing a mold abatement, where they're doing it, what they're doing, how much, what's the block number, the lot number, and yada yada yada. And then they finish, and then the assessor has to come in and say, yes, they did it, it's done, and the problem is fixed, and they have to file with DEP a form that says it was done. So there is a process for New York City to provide some oversight on mold abatement and for enforcing mold cleanups and fixing water damage for landlords to get that done. So a little bit about where mold grows and how to sample. We'll get this part here. So molds grow wherever there's moisture, as I said, and sometimes it can grow. This is a duct inside the duct where the air conditioning system was oversized. It's a long story, but actually you can have an air conditioning fan unit that is too powerful. That's kind of why you want to have an engineer design your mechanical system. You want to design the amount of cooling capacity to be just right. It's you don't want to over design it, because what can happen with an air conditioning system, these central systems, is if it's too much cooling capacity, in other words, that you might find that for one floor of a building you need 6 tons of cooling capacity, and the engineer says, well, you know, what the heck, we'll put in we've got an extra 12-ton unit, we can't seem to sell it, so let's just put that one in there. So now you have twice the cooling capacity for that floor that you really need. And what happens is the air handler will they call it short cycling. It's too able to cool the rooms off. It cools them off so quick, like the room needs to come down 3 degrees from 72 to 60 or 8 or 70. And it's so good, it's so powerful, so much cooling capacity, it can do it in 3 minutes. And what that does is that the system has not run long enough to dehumidify. It's too able to cool the place down, and so you never really dehumidify that air. So if you design a cooling system to be just the right capacity, it will have to run for a while to cool the room off. It might need to run for 15 minutes or 20 minutes before the compressor kicks back off. And what that allows for is a gradual cooling and a much better dehumidification. The air keeps getting dehumidified through the coil. And so over designing it. And what happened here is it was very cold air and it was humid air. And it actually, that's the lining. The second thing they did badly in this building is they have a lining of their ducts right inside the ducts. Nobody does that and should be doing that anymore. The ducts should be insulated on the outside. Duct lining is something to avoid. So here we have, this was a fairly new building, and all the ducts are lined, and they all grew out mold, and so now they have to, in a 3-year-old building, they have to tear out all these ducts, every duct, and put in new duct. You can't imagine the costs for doing that. You have to cut open walls and the ceilings and everything. But so it can grow in the ventilation system, and that's hard to spot. This got picked up by the mechanic, the engineer, who was just doing their regular, inspections inside the system, right? You have access to various parts of the ductwork, and he was smart enough to know that this is not a very good situation. You want your ventilation system to look like that, right? Where it's nice, clean, there's no pooled water. That's a coil, right? So the way this works is I turn there's a fan pulling this air into your building, and that air is going through this. This is a coil. We call it a coil. It's got in the air conditioning season, this has got really cold water flowing through these, almost like a car radiator. But there's all little holes, and the air can go through it, but the water is contained. But this coil will get to be about 40 degrees in the air conditioning system. That's what we call it, chilled water running through it. And when that hot, humid air from outside comes through this coil, it generates condensation. And here you can see it seems to be doing a pretty good job with dealing with condensation. I'm going to use the laser just because I can. But the pooled water will collect here and drain out, and so it looks really nice. There's no rust. That system's in pretty nice shape. That's a nice, well-maintained, cleaned fan unit. When they aren't well-maintained and clean, you get a lot of rust and slime. Same thing, but this is the fan coil unit. We've all seen these everywhere, but probably if next time you're at a hotel, you'll look at the one you have in your hotel. It's probably right under the window. And here's the fans. They move the air. They pull air up through the filter at the bottom. And then they push that air up through this piece here. Inside this is a coil. And then at the base of this coil in here is a pan that drains the water, and the water is going to drain here into this little drip pan, and then it drains down into a waste line. So everything is good until this clogs, and then you get water dripping over the sides, and that can get under the floor. Uh-oh. Now I'm let's see if we can make this. All right. A different kind of problem here is condensation. And you can sort of see that light looking, you can just barely see it. That's some visible mold. And that's because this is a cooling duct, and it's not insulated, and it's running through a crawl space that's not air conditioned. So you have hot, humid air in the crawl space, you get condensation on the duct, and they decided to paint it, which a duct by itself would be very low mold growth substrate. Mold's not going to like growing on the galvanized metal duct, right? It's just not a good substrate, or on a sheet metal duct. But if you put paint there, the paint becomes paint is latex, which is a protein, and it's going to grow on the paint, which it did. So you do have this situation with what we call humidity mold. This is actually a picture from this past summer. Gene knows where this was from, but this is in a CUNY building. And what happens is if throughout COVID, in this case, throughout the course of, and it may not have even been COVID, but if you're air conditioning a building and you're either short cycling, as I mentioned before, or you have no occupants, so you have no heat load, you can overcool the building, and the system won't dehumidify. It's just overcapacity, right? They've designed the system to have 1,000 people in the building, and you really only have 50, and so the system never stays on long enough, and so you end up getting high humidity. And if the humidity levels can get up over 75% for an extended period, which is about a week or two, you'll actually get mold growing out on things like finished wood. So you see it, this little white fuzzy here, that's hard to see. You have to shine a flashlight down. This is a door. This was a door to in a little lunchroom, and you could not see that. Lots of people went through and looked for it, but unless you have a flashlight and you're kind of careful about how you look, you won't see it. But what you shine the light across it and you see it, and that's all what we call a xerophilic mold. It's a mold that likes very dry conditions, and it's usually Aspergillus penicillioides, or a lot of species of Aspergillus are dry tolerant, this kind of dry tolerant mold. So it's a humidity kind of mold, and we saw a lot of it with COVID when everybody was home and not going to their offices, and when they were still air conditioning in buildings, when the heat load was just much smaller, and so we had too much cooling capacity. That was the underside of a desk, a table. You can see just from there's no water staining. It never got wet, but you see the little spots where an Aspergillus, xerophilic Aspergillus, will always have this white color, and that's what happened. You can see there's even a little one here growing there, and these can really release a lot of spores into the air. It even grew on some of the furniture and the chairs. But this is more of the flood the building and really get them wet. And this is what happens when you flood a building. Here's a sheetrock wall, and the water always goes to the bottom of the wall. And so, but then, of course, in most of our buildings, we put vinyl along the bottom of a wall. That's the cove base. And so this part of the wall never dries. So this part of the wall is dried up. It got flooded, the wall got wet, but it dried. But the vinyl trapped the water down here, and then you see it grew mold all behind the vinyl. We would now have to cut the bottom foot. And as I said, there's much more mold inside the wall cavity than there is going to be there. And that's what you see. You open up the wall, cut that sheetrock out, and you can see how it grew much more inside the wall cavity. And if there's an outlet, electrical outlet somewhere higher up this wall, you'll get some air moving with spores out through it. Who can see the transcript? Oh, okay. Condensation. This is on a chilled water line. We see this in the summer. Sometimes buildings will upgrade their air conditioning. They put new chillers in. They make their chilled water goes from being 45 degrees to 40 degrees, and the insulation no longer is sufficient, and you get a cold. It gets cold on the outer part of the insulation, and that becomes a condensing surface. We call it a cold condensing surface, and then you get enough condensation. I can tell you what's going to be growing here because I've looked at it enough. This is going to be a species that likes cold, really cold, right? Because it's 40 degrees here. And once it gets wet and condensation starts happening, the insulation stops working. So the whole pipe starts to become 40 degrees. And that's what's growing there. It's a type of mold that loves it cold. This is condensation in an attic. Somebody hooked up the bathroom exhaust, never ran it outside. They just dumped all that bathroom exhaust into the attic, and it condensed out on the deck of the roof and grew mold up there. Usually it doesn't present a huge risk to the occupants because the air from your attic doesn't really move much down into your house. The air tends to move the other way around. It tends to move from your house up to the attic. Sometimes it's, as I said, mold is hidden. And so this was a plaster ceiling, and we had to deal with the dogmatic molds never grow in plastic crowd. But if you take the plaster out of the way, you see there's what we call backer board, and that's like a sheetrock board behind the plaster, which it can grow very nicely on, and it can grow very well on the wood lath behind some plaster. So yes, plaster is less supportive of mold growth, but please, no dogma. I don't like it when I hear people saying that mold doesn't grow on plaster. It just is wrong, and they shouldn't say that because they're telling everybody I don't know what I'm talking about. And I usually say to them, I'd like to agree with you, but that would just make both of us wrong. All right. But vinyl wallpaper traps water, right? So you can see in this scenario, you would not see any visible mold. It's all behind the vinyl. And this is a building in Puerto Rico. And what would happen is every time it rained, it's a concrete, this wall is concrete with a skim coat of plaster. But when it rains in Puerto Rico, the sun comes out shortly thereafter and really cooks the side of the wall. It really heats it up. So it drives the moisture through concrete into the building. So when you have rain on the outside of your building and a hot day, the drying is going to dry into the building. It's going to, the driving force is going to push the moisture in. And you need to design that, engineers design these walls so that moisture won't do that. You have vapor barriers and things. You can see how it wicked into the sheetrock that was touching the wall. And then the water got in behind the vinyl, and people in the office, this was a federal Social Security office. They were saying, oh, everybody's getting asthma attacks, and we're all allergic, and it smells musty in here, but nobody can figure out where the mold is. And that's my hand pulling that wallpaper away with my old watch. And that's a long time ago, this picture, but that's really kind of an interesting, case study where the mold is not visible. You have to sometimes look for it and move stuff out of the way. So it can be tricky. Here's a floor right under a vinyl tile, and they had some steam leak at some point, and they fixed the steam leak, but it warped up the floor, and we pulled up a couple of tiles, and it's hard to see, but there's a little bit of a greenish look here, like this powder. See, there's a little bit of greenish stuff right there. And we take a tape lift from there, and it was all penicillium growing under. And what happens is, if you're getting moisture coming up from under the floor, it can't get through the vinyl. The vinyl is a very good vapor barrier, so it just gets trapped in the wood. So when we deal with wet buildings and flooded buildings, it's really important to try to figure out where do you have these kind of layers of materials that are going to trap water, because you'll never get them to dry, and you have to figure out how to. You might have to remove some layers of vinyl or other vapor barriers if you're going to get the rest of the stuff to dry. Otherwise, you'll start getting walls that look like that. These are these kinds of molds on a sheetrock wall. These are what we call hydrophilic. They need a lot of water. They have very high requirements for water. So that we have three kinds of mold we call xerophilic. They don't like much water at all. They like it dry. Then we have mesophilic. They like the medium amount of water. And then you have hydrophilic, and they like a lot of water. So when I go back to doing my high dust samples, and I vacuum dust on a shelf, and I culture that out, and I find that I have mostly hydrophilic species, I can say that there's something really wet in this building that's releasing spores. Or if I have all xerophilic, I can say there's some humidity related mold here. It's not always so easy, because sometimes if you're going to culture out molds, you have to actually promote. If you want xerophilic molds to grow, you got to grow it in a somewhat dry environment. But for the people who have unlimited funding, you can culture it out in 3 different culture media and look for the xerophilic, the mesophilic, and the hydrophilic. You don't really need to do that. Sometimes I'll do two different media. Xerophilic would be DG18 agar, and mesophilic is MEA. And by doing those two, you can tell what's going on by which one really gets a lot of mold growth. You can sort of get a sense of what's happening. So abatement. I know we're running low on time, so I want to get us through cleanup. And here, with what we have to do, like those sheetrock walls, we've got to cut away the sheetrock and replace it. You can't clean sheetrock. Can't clean this kind of wood when it's gotten moldy. Yeah, it's got some. I think I have some different. Oops. Oh, so here we go. So different kind of samples. I think we talked about this. We have bulks, swabs. A swab sample would be more of a Q-tip type of sample, and you can either culture it or have them look at it directly. Tape lifts As I said, you take a sample of whatever that discolored looking stuff is, put it on a microscope slide, really easy to look at. Anyone that has a microscope that's 600 times or better, it's just an optical microscope. Molds have color, so you don't have to stain them. They're really not hard to do these kinds of analysis. And you can do culture plate sampling where you actually get the species, or you can do spore trap sampling where you're just looking for spores and you only really can identify those to the genus. And the vacuuming, as I mentioned, I take for vacuuming samples, there are different products you can buy. I just use a 37 millimeter cassette that you would use for asbestos or lead sampling. And I put it on a sampling pump and just vacuum a surface and collect all that dust and measure the area and then culture that. And we'll, so there's a typical agar plate for doing air sampling cultures. It's a Anderson sampler, and it will shoot air through an impaction device, and they'll make 400 impaction points on that agar plate. It's a short sample, it's 2 or 3 minutes, but the lab can tell you the species that are in the air, and that compare that to the outside air, and you can start to get a sense of whether something's going on. That's a spore trap. Basically, it's just a microscope slide in there with some clear, sticky grease on it, and it's a slit in that collector, and the air goes through that slit very rapid pace, and then it turns at a right angle, and all the spores impact the grease, and then the lab mounts that up under the microscope and counts the spores, and they can tell that mostly to the genus level. And this is really a 5-minute sample to a 10-minute sample. So it's also not a long air sample. So for the industrial hygienists to know that that has associated with it some pretty large errors. And that's the kind of report you get. I know you probably can't really see it, but you take the outside versus the inside. You see, and this is a place where we have an outside sample, a first floor sample, a second floor. And it's hard to spot, but oh boy, I'm having a hard time even seeing. But you can see just the total spore levels here. They're much higher on the first floor and the second floor. And looking at penasp spores, we're seeing in this case, it's like 20 or 30,000, whereas here we're at 100 and something. So when you see that kind of thing happening where the penasp spore levels are 20,000 and they're 100 or 200 outside, that tells you that there's something going on. But you don't have it, you're not any closer to knowing what's going on, because that just tells you how the air is. It doesn't tell you where it's coming from. And so that was, as I said, my preference is to do tape lifts and microscopic exam. We're looking for those structures, the spores and chains, hyphae, the fruiting structures, conidiphores. When we see those, that's by definition mold growth. So you've nailed it when you've got it on that. So how do you fix this? You have to really deal with the water, and that's what all of the standards require, is address the water intrusion, the water damage. If you don't fix the water, you're not going to fix the mold. You're just going to remove it, and in the course of another month, it'll all grow back on again with whatever material you put back. We use HEPA vacuums, HEPA filters, HEPA filter air scrubbers, HEPA filters on the respirators. There are more guidelines now. There's an IICRC standard for removing mold. So there's lots of guidance published out there on mold abatement. EPA, the rest, all of them have the same basic guidelines. You don't use a lot of chemicals. The goal is not to kill the stuff. The goal is to remove and clean up the spores. This is not an infection risk. I don't care if it's alive or dead. It's going to cause you to still have a kind of inflammatory allergic immune response, whether the spores have been killed or not. So using a lot of chemicals to kill the spores doesn't buy you much in the whole process. We remove the material that has mold growing. We use negative pressure with HEPA filters. We use HEPA vacuum cleaners, and workers generally wear protective equipment like respirators and Tyveks. And that's a pretty good standard there. That's the IICRC. It's an ANSI standard for, it's the standard S520. And that, if you want that, that you have to pay that. That's like an NFPA standard. It's probably about $80 or $90. And there's the New York State has their own regulation. So New York State has a mold regulation. It mainly covers the licensing of mold contractors and assessors. This is what we don't want people to do. And that there you see the landlord came in and just demolished the wall and left debris all over. Made a mess. Duct cleaning can sometimes help if you have any kind of thoughts that you might have gotten a lot of spores into the duct system. There are duct cleaning contractors. This is a school gym in Rockaway. This was in 2012 after Sandy, right? And so everything got so wet where they just pulled up the whole floor, and you see they have these HEPA negative air units just pulling all the air out, creating negative pressure. But you see, this was a gym floor here, and this was a few weeks after the flood. You can see the concrete is still wet. So it's really hard to get it to dry and so we have to pull all that stuff that was warped and moldy. There's a typical prep of an area, put plastic sheeting on floors to and protect contents, right? Put barriers up so that you have some containment of the dust. And then when you're done, this is kind of where what you end up with is you've removed the sheetrock that had mold growing on it. And you see it's all off of this wall. And this allows you now to look inside the wall cavity, make sure that this other side of the wall, this was a bathroom on the other side. And so it was very helpful. Nothing was growing on the back side. So we knew we didn't have to tear down their ceramic wall tiles. And we protect the contents. We usually remove the contents, wrap them up and get them out of the way. But again, mold is hidden, and I know I'm running out of time here, but we'll just so this is the same location here. The contractor came in and took the lower ceiling down that had mold on it, but didn't take out this foam panels. I wrote a specification in this project and said, take it all out, go all the way to the framing. I don't want to see anything. So they didn't. So when we took out the panels they left in place, that's what it looked like. It was moldy above. And they did the same thing with the floor. Again, we said, take up the wood floor, take it all the way up, go down to the concrete. They did not. So when we picked up a piece of the sheathing, you see it's all moldy underneath. It grows really well where the water gets trapped. So the water doesn't necessarily get trapped on top. It gets trapped under. And this flood happened in like 2018. We pulled this up in 2019 because it was a fight with the landlord, and the landlord didn't want to do it. And we just went back and forth with lawyers, and everybody gets involved. We spend all these days in court to fight over doing something that should have been done. So they did it, but they did it very begrudgingly. So of course they didn't want to do a very good job. And so I showed up after they were done, and I pulled up this one piece and said, good job, guys. You left all this nice mold underneath the plywood, and it was still wet under there. A year after the flood, it was still wet under there. So water won't go away by itself. You have to dry it sometimes. There you go. That was where I took that plywood up. You see, that's all mold growing here. And it's what, 43%. A reading over 15% would be considered too wet. So there we want to remove the mold and then build back. It's negative air. They had to take up a finished floor. And then when they took the finished floor up, the plywood was also moldy. So we took up the plywood. And there's the inside of work area. This is typical. The guy's got a Tyvek suit on and a respirator on, and they're cutting the lower 3 feet of sheetrock. There's a negative air tube. They're running in negative air. Then they're going to pull up all the carpet tiles. Everything gets bagged. Moldy waste is not regulated. It can go into any dumpster. You just have to bag it all up, seal the bags, and put it out in the dumpster. It's regular C&D waste. But you can see when you cut open the wall that these walls look pretty good. But when you actually cut them open, you see the mold is all inside. It's a vinyl trapped, in this case, trapped a lot of moisture. So just more of the same, cutting out the lower sheetrock. And then in this case, this is a school in, this was actually right next to LaGuardia Community College. This is a school over there, and they had constant flooding. It's one of those things you get in Queens when the storm drains back up a lot. And so they're always flooding. So we basically had a contractor came in set up a mold project. That was the last, that other picture you saw. Come on, where were you? That one. That's where they cut it all out. And then we had, because it had a likelihood of flooding again, we put this stuff called Den's armor, which is a type of sheetrock that has no paper. So it's nothing is fully mold resistant, but it's pretty mold resistant. It's more forgiving. So we put that back. There you go. Den's armor plus moisture and mold resistant. It's a fiberglass gypsum panels. So, but the mold will grow in the paint, and it can grow in, if you have a flood and it's really a gunky water, it's got a lot of organic material in it, like storm water, then it can grow in anything. It's going to grow in the soil and gunk, that's organic junk that's in the water. So the water can bring with it, if it's dirty water, it can bring with it all the nutrients that the mold needs. It doesn't matter if it's there's no paper or organic substrate. More information, you can go to New York City Health Department guidelines, but there's probably a lot more out there. I would recommend you stick to the the more tried and true guidance, the AIHA, the EPA, Health Department, Department of Labor, and IICRC, and NIOSH. You stick to those, you're in better shape, and don't get too many people get into reading stuff in the internet that's wrong, just completely wrong. So anyhow, that is I think I've kind of run out of time, but I guess if people have questions, I could take some questions. I don't know.
We do have some questions.
How did I figure?
All right. So let me ask a few that are more related to concerns people have at home and mostly related to cleaning. There was one about the best way to clean dust at home so that it doesn't just end up in your carpeting.
Yeah, well, I think it's probably, if you have a lot of carpeting in your house, it's not a bad thing. A lot of people have a policy of taking their shoes off when they come in. And I think that's not a bad plan if you have a lot of carpeting, because you're going to track a lot of stuff on the carpet. It's hard to clean. You can HEPA vacuum the carpet, but ultimately I think you need to shampoo, extraction clean it probably once a year, unless it's a really high traffic area. And that is and it's probably better to do it professionally, where you bring in like Stanley Steamer or one of those companies, and they'll shampoo the carpet, and they have really good extractors, and the idea is that you have to suck all that water out after they shampooed it so it's dry. If you rent those extraction cleaners, they may not have enough oomph to really get the water back out, and so you're not getting the dirt. It's better done professionally once a year. So not hot water, shampoo with extraction cleaning once a year for carpets.
What about just the dusting? I mean, is dry dusting going to move it around, or is wet dusting better? Or what do you think about that?
Dry dusting mostly works okay. I don't think you have to. Damp wiping is not a bad thing, though. And HEPA, I think people should have a HEPA vacuum cleaner today, and not, you're better off with something that has a good filter on it, and just vacuum dust or damp wipe. It's probably a little better than dry dusting.
So then we had a couple of questions about people with bathroom issues. So cleaning black mold on bathroom tile and on a sealant around a tub. So people have tried bleach, tried vinegar, they don't work. Any suggestions there?
Right. So what's happening there is, it grows on, it won't grow on the ceramic and on the grout, but it's growing on all of the other junk, the skin cells, the soap, all the stuff that gets into the grout. If you clean it with a good Mildex or bleach, you will kill all and remove all the mold, but what you're seeing, that discoloration that remains, is probably just because molds don't, you know, they digest things externally. So as I said before, they will break something down. So when you kill off all the molds, you're still left with their damage. It's sort of, they've already pre-digested that surface, that caulk and that grout. And so you'll never make it look new again. It gets discolored. Also be wary if you're getting, if it seems to keep coming back rapidly, it could be you're starting to get moisture behind the tiles and it's staying wet back there. So if you clean the tiles and they just mold right up in a week later, you might want to start looking into the possibility that you know, I don't know how old that tile wall is, but you know, they don't last forever, and at some point you might have a point where you have to retile it. But you know, that's today when you build a shower or a bathtub, they would put a cement board. They don't use sheetrock anymore. So if you have a bathroom installed in the last 10 or 15 years, they would put cement board and then put the tiles on the cement board. You won't get mold growing on that. So, but it can grow on the grout and the tiles. It should clean off. If it's still discolored, it's often because it's digested that stuff. You could regrout it and that might do it for you.
Okay. Thank you. Now, so we have some all important questions about our CUNY buildings and how we put pressure on administration, what legal recourse we have. So I'll just kind of try to summarize some of those. There was a question about how often, if at all, CUNY has to test buildings for mold and whether there's any legal requirements about that. And then how can we as a union promote more testing and remediation, and what legal recourse do we have to get administration to actually do something about this problem?
Well, there are no regulations necessarily that like an OSHA regulation, but I think if you are you know, if you have water damage and you've identified mold growing on things, I would start with just going to the administration first and just say, look, this is moldy. People are getting sick. You need to take care of this. And obviously, I'm sure you have. And if you're still not getting anywhere, I think you can get PESH might come in, especially if it's visible mold. But there are no regulations. I think PESH would issue a citation to correct a problem if they found water damage and mold growing. But there's not a lot you can do to compel them necessarily to fix a mold problem. The regulations are pretty weak. There is for housing, but not so much for the other sectors, buildings.
Right. So I see some of the people who had those kinds of questions are still here. And if anyone wants to raise their hand, I think we're a small enough group now, we could probably. Yeah, Megan. Oh, you have to unmute, Megan.
Sorry, we've had an ongoing issue at City Tech, which is why I was asking about recourse. And one of the things I've been trying to figure out is, in addition to, we had documented it, they kind of said, I think what you were saying, they said that they had remediated the issue, but won't give us any information. And my understanding is I looked through the DASNY regulations that they have to, if they do agree remediate, they're supposed to provide reports, but they're telling us that they don't have any report. So it's hard to figure out even if they actually did do anything and what our rights are even about getting documentation about that kind of thing. And it was pretty widespread in the building. We had photos, and it's been an ongoing issue that comes back. So probably it is a ventilation issue. And I know it's I've dealt with it in my apartment, so I know the regulations there, but it seems like it's much harder to actually push or get information that we need at City Tech. So if you have any thoughts or suggestions, I would love to hear.
Well, I think you're probably right. If it's coming back every summer and it starts to form on all the ventilation outlets, the ceiling diffusers, or on other objects, it's a humidity kind of mold growth. And they can go and clean it, but ultimately there's got to be an engineering fix. If it's really the HVAC that's causing this, and they need to get some mechanical engineers in there to actually figure out why they're getting so much humidity and correct that problem. Otherwise, it's just going to keep coming back and coming back. Getting the New York City regulations are all entirely focused on residential, and so I don't think you can look at local law 55, but I think that's entirely residential. I don't think. I think they're not able. The city is not really able to regulate workplace safety because that comes under OSHA's domain and PESH. If you're still getting mold growing and you can see it growing on things, all you can try to do is put a PESH complaint in and have a PESH inspector come and cite them. That would be the only regulatory process. And maybe the health department, I don't know. I think the city agencies would sort of try to stay away from workplace issues because it becomes this jurisdictional stuff. PESH and OSHA with OSHA.
We actually have had for some other issues. We have had some success in having people call into 311.
Yeah, you might get you'd probably get a health department person coming in.
Yeah, they will. I mean, it's not a solution. It's like another pressure point. I mean,
Right.
Essentially what we find is we have to harass CUNY from several different angles before anything gets done.
Yeah.
And then unfortunately, it's like what you said earlier, Ed, that they'll clean off, wipe off the mold, but not fix the problem, which it's City Tech, there is a structure, infrastructure problem.
Was that the building we looked at, City Tech? That's not the one in.
It's
Brooklyn.
Affiliated.
Oh,
It wasn't, it's different.
Okay.
It's nearby.
But it's the same problem. It's this
Yeah.
UP system running amok. Yeah, and that's a hard one to fix. And when we looked at the one out there in Brooklyn, they were totally convinced. They sat down at our meeting and said, there is no mold growth in this building. And we went around as a walk around, and I think in about 15 minutes found them lots of mold for them to. So in their defense, they relied on their contractor who was not necessarily, there's just such a range of people who are, some are really good at doing this and some are not so good at it.
But we need to pursue the situation.
We need to get people
It's
better
City
at
Tech.
this. Yes. Yeah.
Any other questions?
Keep plugging, because I work with the UFT 2 days a week, and we have dealt with New York City schools, and I have been pushing mold in the schools for 25 years, and after all of these years, we've really finally gotten to a point where that we get no pushback. If we come in and say we want this fixed and done, they do it. Just keep plugging away. You'll get them there. It's a long process.
Okay, I think we've come to the end of our time. Thank you to Ed.
All right.
Thank you to all of you who stayed for a long time after a long day. And took this in. And it's a lot to take in. But it is a lot to take in. So you can watch the recording. So
Yeah.
that will be available, probably not this week because of all the stuff that's going on, but next week, and we'll send out a notice. So
And maybe someday we'll do on responding to floods, because I never did get into responding to floods, how you're supposed to do
they
it.
happen.
It
Yeah.
happens.
Okay. Thank you all and have a wonderful evening.
Thank
All
you,
right.
everyone.
Very
Thank
good.
you, Ed, especially for all your
Thank you.
input.
Thank you, Ed.
See ya.
Bye all.
Bye.